Welcome to the second algorithm and data structures for sequences session, which is also the last talk session of JOBIM for this year. We have three in advance nice talks from Anne Guichard, Pierre Maurice, and Cédric Chauve. The first talk will be given by Anne Guichard, and the title is MTG-Link, Filling Gaps in Draft Genome Assemblies with Linked Reads Data. Let's see, Anne, can you share your screen? Excellent.
Okay. Okay.
So please go ahead.
Okay, so good morning, everyone. So I'm Anne Guichard. I'm working in the GenScale team within the INRI Institute. And today we'll present you MTG-Link, which is a pipeline for filling gaps in draft genome assemblies with linked read data. So I assume some of you may not really know what is linked read sequencing technology. So I will just give you a short introduction about this sequencing technology, and I just put here a figure of the 10x genomics technology, but basically the concept is roughly the same for all linked read technology. And so in this technology, long reads are generated from long DNA molecules, and every short reads that have been sequenced from the same long DNA molecule are tagged with a specific molecular barcodes. So in this figure, we have in gray, we have the short reads, and had they all come from the same long DNA molecule, they all have the same yellow barcode. So these non-contiguous reads are referred to as linked reads. And by linking the short reads together via a short barcode, linked read sequencing technology combines the advantage of ease and accuracy of short read sequencing with the long-range information of long read sequencing. So it's possible to reconstruct single molecules with some accuracy and at a low cost. And for this reason, linked read technology can be applied to many different applications, such as de novo genome assembly, haplotype identification, and structural variant coding. But we are interested in de novo genome assembly here more specifically, and we know that there exist some tools for genome assembly and also scaffolding using linked read data. But the problem with these tools is that quite often when they merge contigs into scaffold, they put ends between the contigs. And so actually they lead to incomplete assembly with some gaps left in the assembly. So there is a need for gap filling tools, for efficient gap filling tools, and several tools exist for closing gap with short read data, but to our knowledge, none uses the long range information of the linked read data. So the aim of our work was to develop a gap filling tool that uses linked read data. So we developed MTG-Link, which is a gap filling pipeline for draft genome assemblies dedicated to linked read data. And MTG-Link is composed of 3 steps. So the first step is a read subsampling step. Then there is a local assembly step, followed by a qualitative evaluation step. And the main feature of MTG-Link is that it takes advantage of the linked read barcode information to get a subsample of reads of interest for the local assembly of each gap. So how does it work? So MTG-Link takes as input a set of linked reads, a GFA file with gap coordinates, and an index PUM file. And as I said, the first step is the read subsampling step. And this step is used to keep only the reads of interest for the gap filling process. So in this step, for example, if we have 2 sequences and a gap between them, we define gap flanking sequences that appear in blue here, and we call them chunk. And so the pipeline will extract the barcodes that are observed in chunk region, and it does this using the tool LRZ, which is also a tool developed in the GenScale team by Pierre Maurice, who will talk after me. And this tool enables to process linked read data. So we used it to extract the barcodes mapping on chunk regions. And then we do the union of the barcodes. And so to increase specificity, we can decide to keep only the barcodes for which the number of occurrences in the union set is larger than a defined parameter. And then we extract all the linked reads that have the barcodes observed in the union set. So even if a read doesn't map on the chunk region, if it has a barcode that is observed in the union set, it will be extracted. So the goal of this step is to get a subsample of reads for the local assembly of each gap. So instead of using the whole set of reads, we will just use a subsample of reads of interest. And so it will reduce the complexity of the assembly graph and the running time. And then this subsample of reads is used in the local assembly. So the goal of this step is to find a path between a source sequence and a target sequence surrounding a gap. And two assembly algorithms can be used during this step. So the first one is the deep-root graph algorithm, or DBG. And the second one is the iterative with overlap algorithm, or IRO. So the DBG algorithm is performed with the field module of the software Mind the Gap, which is also a tool developed in the JanScale team. And it relies on a deep-root graph data structures to represent the input read sequences. And so in this algorithm, we search for exact overlaps between k-mers, and the overlap size is fixed and small. So it's well adapted to high coverage data. And I also implemented the IRO algorithm, which is based on on-the-fly computations of read overlaps and iterative extension of the current assembly sequence. And the main difference with the DBG algorithm is that in the IRO algorithm, we search for inexact overlaps between reads, and the overlap size is variable. So actually, the IRO algorithm is well adapted to low coverage data, and as we have variable overlap sizes, it can easily adapt to the sequencing depth. So these algorithms enable to get gap fill sequences, and each gap fill sequence obtained is evaluated during the qualitative evaluation step. And to be able to evaluate the gap fill sequence, we have extended the gaps on both sides by a certain number of base pairs, so it corresponds to the blue shading here. And so it's so actually the local assembly step was performed between the sequences surrounding the extended gap. So not the actual gap, but the extended gap. And so we can compare the gap fill sequence obtained to the sequences corresponding to the extension of the gap. And based on this comparison, MTGlink assigns a quality score, and only the gap fill sequences with a good quality score are returned. So this qualitative evaluation step enables to infer the quality of the gap fill sequences, and it might help filtering out putative erroneous sequences. And after this step, MTGlink output the results in an assembly graph in a GFA format, and the set of gap fill sequences are returned in a FASTA file. So MTGlink uses also a trivial parallelization scheme where each gap is processed independently from the other, so it enables to save time when we give it multiple gaps to fill. And MTGlink is fully available on GitHub and also as a Bioconda package. So I just put the links here if you want to test it. And I just want to mention also that it's still in development. So if you have any problem using it, just please contact me. So then I tested MTGlink for its efficiency. And to do so, I used a dataset of real 10x reads from the butterfly Lyconus numata genome. And for 4 different gap sizes, I simulated 57 gaps in a supernova assembly obtained from this dataset. And then I tried to fit these gaps with MTGlink using either the DBG algorithm or the IRO algorithm. And I compared the results obtained with MTGlink to those obtained with the short-read local assembler Mind the Gap. So as we are working on simulated gaps here, we know what the actual gap sequence is. So I will be calling it the reference sequence. And so we can easily evaluate the quality of the results just by performing Blasten alignment of each obtained gap fill sequence to the reference sequence. So in the figure here in the x-axis, we have the different gap filling tools that were used to fill the 57 simulated gaps. And then for each pair, we have a color code. So in blue, we have what we call the successful gap fillings. So this corresponds to the gap fill sequences having more than 85% identity and coverage with the reference sequence. In yellow, we have the poor gap fillings. So these are those having less than 85% identity and coverage with the reference sequence. And in gray, we have the no gap fillings. So this corresponds to the gap for which MTGlink didn't return any solution. So actually, it corresponds to the gap for which no gap fill sequence with a good quality score was found. And if we compare MTGlink DBG with Mind the Gap, as the DBG algorithm in MTGlink uses Mind the Gap to perform its local assembly step, this step is exactly the same between both tools. So actually, the main difference between these two tools relies mainly on the read subsampling step. And we can see that MTGlink DBG outperforms Mind the Gap because it returns much more successful gap fillings. So actually, we can see here that the read subsampling step greatly improves the quality of the gap filling. We also compared MTGlink to the short-read gap filler or gap closer. And it appears that gap closer gives pretty bad results because only 24% of base pair were filled and 0 gap was entirely filled among the 57 input gaps. And so this is quite promising for us because it shows that the gap filling is more successful when we use linked with data with MTGlink when compared to using short-read data. So then I tested the quality of the results obtained for 4 different gap sizes, and we can see that for 1 and 5 kbp gap size, more than half gaps were successfully filled with MTGlink DBG. So actually, the gap filling is mostly successful for small gaps, but it is more difficult to close larger gaps of 10 or 20 kbp. And this may be explained by the fact that there is a higher probability of repeated regions in larger gaps that makes the gap filling more complex. But we can still see that MTGlink DBG gives a greater added value for large gaps when compared to mind the gaps. So this is quite good results. Then if we compare both assembly algorithms of MTGlink, we can see that the IRO algorithm does not give as good results as the DBG one, but it still gives better results than mind the gap. So we can wonder why, in this case, the DBG algorithm gives better results than the IRO algorithm. And the first, maybe it's because in the IRO algorithm, we use a heuristic, so we don't do an exhaustive search of all possible paths in the assembly graph. And also in this dataset, we have pretty good coverage, at least for linked with data. So the DBG algorithm succeeded in filling many gaps in this dataset, but maybe if we test MTGlink on other datasets with lower coverage, the IRO algorithm may actually perform better. And besides, we also observed that some gaps that were not successfully filled with the DBG algorithm were actually successfully filled with the IRO algorithm. So actually, both DBG and IRO algorithm may be complementary, and you can prefer to choose one over the other depending on your input data. So then we wanted to see the impact of the record selection on the quality of the gap filling performed by MTGlink. And interestingly, we can see that when we have a very small number of records, so less than 500, there is a higher probability of not being able to fill the gaps. But as we can see here, a higher number of records does not guarantee that the gap will be successfully filled. And indeed, when we try to increase the chunk size, and so consequently, we were getting a larger number of records, we didn't improve the gap filling. And more precisely, we observed that the gap fillings that are labeled as no gap filling, but that have a number of records higher than 500, are those for which MTGlink finds a solution, but with a bad quality score. So we have seen that MTGlink performs pretty well on simulated data. So then we wanted to see how it performs on real data. And for this, we have worked on the SuperGen project, which focuses on the reconstruction of the SuperGen locus in the genome of the butterfly Heliconus numata. And so this locus is a locus of biological interest in Heliconus numata because it controls a mimetic wing color pattern and is subject to rearrangement polymorphism, as we can see here. But the problem is that the supernova assemblies obtained from this dataset are incomplete, and there are still some gaps left in the assembly, as we can see in the purple column. So we tried to fill these gaps using MTGlink with the DBG algorithm. And we got promising results because we were able to fill 45 gaps out of the 59 initial gaps. And besides, for 3 individuals, we were able to reconstruct the SuperGen locus as a single scaffold, as we can see here in the last column. So to conclude, MTGlink is a robust gap filling pipeline dedicated to linked with data. Its main feature is to get a subsample of reads of interest for the local assembly step. And we have shown that this read subsampling step quickly improves the quality of the gap filling. Besides, MTGlink is mostly successful for small gaps, but the gap filling tends to be more complex for large gaps, for which there is a higher probability of repeated regions. But overall, we still got promising results because we were able to get better genome assemblies on the SuperGen project. Besides, MTGlink here was applied on 10x linked tweet data, but of course it's compatible with other linked tweet sequencing technologies such as haplotagging, STLFR, or TELSEC. And then there are still some improvements that need to be done on the MTG-Link pipelines, as for example, getting a gap size estimation using the barcode information. We also want to be able, we also have to continue to compare MTG-Link to other short-read gap fillers. So we already compared it to Minder Gap and Gap Closer, but it would be great to compare it to other short-read gap filler that uses a different algorithm. We also want to apply to other real data sets from other linked tweet sequencing technologies, and we already started to apply it on haplotagging real data to get the breakpoint assembly of some inversions. So to conclude, I just want to thank all my colleagues from the INRAE and RIA institutes that helped me with this work, and also people from the CIFI lab from providing us real data for the SuperGen project. And I also want to thank the ITN Ignite for financing this project. Thank you for your attention.
Thanks. This talk was perfectly on time. Let's take a few questions. So to ask questions, you can either write it on the Q&A or raise your hand, depending on your preference. I can also already see two questions written. If you actually want to talk and tell your question live, please raise your hand. Otherwise, I will just read the question from the Q&A. So Matthias says, nice talk. Matthias Niki says, nice talk. Barcodes are not always unique. How do you deal with wrong routes?
Well, yes, so the problem is that in this technology, several different DNA molecules can share the same barcode indeed. And so we have false barcodes, wrong routes, it says. But in the error algorithm, we have several filters. So actually, when we do overlaps between reads, we want to have at least a certain number of reads sharing the same overlaps to be able to extend the current assembly sequence, and it's the same in the DBG algorithm. So we add some filters that I didn't present here, but these filters enable to say, okay, so if I don't know, 5 reads share the same extension, okay, I will take it into account. But if only one read present this extension, I will not take it into account.
Okay, thanks. Matthias has another question, but I will let you answer it by chat after. I will take the benefit of asking another question from an anonymous attendee who says, thanks for the talk. What scaffolding tool would you advise to use before MTG-Link using PacBio or Nanopore long reads?
So I don't really know. So the results I presented here were used on supernova assembly. So we used the supernova assembler. But if you have any linked reads, you can also use the ARC scaffolding tool. We didn't try to perform gap filling on genome assembled with long reads. So I don't know how it performs. I think it should perform well, but yeah, we didn't test different scaffolding tools. We just use assemblies, genome assembled with supernova and scaffolding with ARCs.
Thanks. Any other questions from the audience? I will take the opportunity to ask a quick question. So the elephant in the Zoom room is that 10x technology no longer deliver kits for genome assembly. So what is your thought about doing long read sequencing for the butterfly genomes that you had?
Well, so yes, so 10x Genomics does not do genome assembly anymore, but there are other, as I said, there are other linked read sequencing technologies. So, for example, there is haplotagging, which is pretty fast, and it doesn't. It's quite cheap. So, linked read sequencing technology is not dead. There are other sequencing technology, and people that works on the We have some colleagues in Montpellier that work on the SuperGen project, and I think they were sequencing the genome with long-read data. So actually they are working on this, and we'll see how it performs. We don't have the results yet, but maybe yes, using long-read data could give, I assume it could give better genome assemblies, but maybe as it's quite expensive and it's still error-prone, there will still be a need for gap filling, and linked read and gap filling with a linked read is ideal when you don't have a high budget.
Yeah, thanks. There is one last question from the Q&A, so I will just take the opportunity to ask it. So it's from Julie Orjuela, who asked a related question to the one before. So can MTG-Link be used after hybrid scaffolding, using, for instance, optical maps on nanopore? In this case, is it necessary to have Illumina to fill in the gaps? Yes.
So yeah, it's quite related. I think, yeah, it can, well, actually, MTG-Link it could be applied on any genome assembly, and it just needs linked reads to be able to work. So if you have just Illumina to fill the gap, it will not work, because in the Illumina reads, you don't have the barcode information. And the main feature of MTG-Link is that it uses the barcode information to be efficient and to work well. So it's not possible to use Illumina reads, but you can perform the gapping on any genome assembly as long as you have a link to data to of any kind of technology to be able to fill the gaps.
Yeah, sounds good. Okay, I will let you answer Matthias' question on the Q&A directly. So thanks again.
Thanks.
Thanks.